Hello, my name is Taichi Suzuki. I'm an assistant professor at Arizona State University, and I'm very excited to share our unpublished work from my postdoc working with Ruth Ley at the Max Planck Institute in Germany. So what is the role of microbiome in plant and animal evolution? And I think this is a very important question because we are surrounded by microbes since the origins of plants and animals. And these host microbial interactions can affect the host biology in many ways. So it can expand the dietary niche, or it can serve as a source or cue of phenotypic plasticity, including traits like physiology, metabolism, response to pathogens, and behavior. And there are more and more studies tackling this question from more theoretical grounds. But we're limited with empirical studies to directly test this question. So how can we test this directly? So artificial selection experiment is one approach. And this is a figure from Miller and Sachs. And I'll walk you through this figure. So when you plant genetically identical seeds into different soil communities, the plants will grow differently. It's known as phenotypic plasticity. So this is nothing surprising. And we can phenotype the traits. And if you're interested in larger plants, we can take these soil communities, inoculate into sterile soil, and we can repeat this process over and over. And this scheme really works. So people have shown that the selection on the microbiome can shift the traits like plant biomass, flowering time, drought, and salt tolerance without any changes in the plant genome. And one of the key here is that these microbial communities are transmitted from one generation to the next. But in general, plants lack these mechanisms of vertical transmission in nature. So it will be interesting to test this in a system that we know that microbes are transmitted across generations. And house mice is a great model to do this because we know there's evidence of vertical transmission in nature and in the lab. And also mammals evolved parental care, which facilitates the transmission of microbes from parents to offspring. And also they're coprophagic, meaning that they eat feces surrounding them. And also germ-free models are available to run these type of experiments. And to provide some ecological context, they show rapid local adaptation. So this is a work from the Nachman lab at UC Berkeley. And if you look at this blue areas, these are subspecies called Mus musculus domesticus, which spread from Europe to around the world with humans in the past couple hundred years. And even in this short time span, these animals exhibit evidence of local adaptation, following patterns like Bergmann's rule and Allen's rule. So animals from higher latitudes show greater body mass, reduced ear and tail length, and increased activity behavior. We also know that microbiome differs by population, both in the field and in the lab. So we can ask, can selection in the microbiome shift the mouse trait over time without host evolution in a controlled setting? So to do this, we first need to identify a mammalian trait that's affected by the microbiome. So we did a simple experiment using these Y-derived inbred lines from New York and Brazil, and do a fecal transplant experiment to germ-free mice, which are inbred and genetically identical. So we used a total of 24 donors and 50 recipients, and we can confirm the successful microbiome transplant. You can see within donors, they have distinct microbiome, and after transplant, there's a minor shift, but they maintain these population differences in the microbiome. So now let's look at the traits. So here's a list of traits that we looked at, and these are the effect sizes between New York and Brazil treatments for donors and recipients. And this is a busy figure, so I'll show you some examples. And this is what I already told you, but animals from colder places show larger body mass, reduced tail length, and increased activity. And this distance travel was measured using this Promethean gauge based on like laser breaks, on beam breaks. So there's no human bias here. So now let's look at the recipient traits. So for body weight, you can see this trend of phenocopying, which was marginally not significant. And when we look at the tail length, we see this significant opposite pattern of phenocopying. And my interpretation is that we know that the microbiome from colder place are better at extracting energy from the diet. So more energy extraction, greater body mass, and greater tail length. So in the donors, these proportional difference in tails might be driven by genetics more than the microbiome. And for the distance traveled, we can see this significant phenocopying. And this persists after accounting for batch effects and body weight at weaning. So overall, compared to these morphological traits, the effect size of activity is larger. So we picked this distance traveled as a trait to select for the selection experiment. And here's the design. And the basic idea is to try to evolve these high latitude microbiomes, which is like mimicking the ancestral state in Europe, and try to evolve the microbiome into more low latitude microbiome, like the Brazil microbiome. So we do a fecal transplant from a single donor into sets of germ-free mice, phenotype them, pick 2 individuals that show the lowest activity, take those feces embeddings into a new set of germ-free mice, and we repeat this for 5 generations. And for the control line, we do the exact same thing in parallel, but do this randomly. And we did this 4 times, involving more than 300 germ-free mice. So the expectation is that control lines will not change the behavior over time, but selection line may decrease the activity if the selection is acting on the microbiome. And here's some results. So here I'm showing you the raw data of distance traveled across time. So you can think of this first round of selection as before selection, because both treatments receive the same microbiome from a single donor. But after selection, for every generation, we see this lower median of distance traveled for selection line compared to control line. So consistent with the prediction, but they were not statistically significant. But after accounting for batch effects and body weight at weaning, we start seeing the significant shifts in behavior only in the later generation, again, consistent with the prediction. And this model comparison also supports this result. So here I'm comparing 2 models predicting distance traveled. And the only difference between these models is this generation, which is information about selection before and after selection. So for example, when we look at the control lines, removing this information about selection doesn't affect the model accuracy. But in the selection lines, removing this information about selection significantly worsens the model, which is the evidence of selection, right? So overall, the effect size was small. We can detect this significant shifts in behavior over time. So now let's look at the microbiome changes. So here, again, showing you a PCOA plot of Bray-Curtis dissimilarity. So you can see, so it's colored by rounds of selection. So you can see a shift of microbiome over time. And these symbols indicate the treatments. And you can see these triangles and circles are overlapping and at the beginning of the experiment, but later on it gets more separated. And you can see this more clearly from this figure. So overall, you can see the increase in dissimilarity over time, which makes sense that the microbiome communities are diverging stochastically over time. But the rate is faster. If you look at this black bars, the rate of divergence is faster between control and selection line compared to within lines. So again, this is evidence of selection on the microbiome, which parallels the shift in host traits. And this is partly driven by the loss of microbial taxa over time. So now let's look at the specific microbial species that associates with distance traveled. So among the 1,400 taxa we tested, these species related to Lactobacillus genus negatively correlates with distance traveled. So when we look at the raw data, you can see more Lactobacillus animals are more calm. And you can see this increase in lactobacillus only in the selection line. And again, this is consistent with the phenotypic data. And when we look at the metabolites, indolactic acid correlates with distance traveled and genus Lactobacillus. And this is further supported from the literature that we know, so tryptophan only comes from the diet, and lactobacillus can break this down and increase indolactic acid. And this is also shown to be reduced in hyperactive autism patients. So the direction, there's some parallels between humans and mice. So in summary, what I showed you today is that mice that show high activity coming to a new environment where there's a selection for low activity, within 5 generations, these animals can shift the trait in response to selection through the microbiome without any changes in the host genome. So I think this is pretty significant that it provides a novel microbiome mechanism of host adaptive plasticity, which can be transmitted through across generation and also within generation through horizontal transmission. So this is a bit Lamarckian mechanism. And also the results imply that the selections acting on the community level function, so selection above the organismal level, and also have some practical application where culturable and unculturable microbes can be engineered for a desired purpose in mammals. So coming back to this main question, I see the microbiome as a modifier of adaptive landscape, which can affect the rates and trajectories of evolution. And compared to more established systems, like insect and their endosymbionts, what I showed you today, that even a complex microbiome can respond to selection and affect the host phenotype, and this can be transmitted vertically or horizontally. So I think this is a very exciting field to further explore the role of these microbiomes in the ecology and evolution of animals and plants. So for that, I'd like to thank the Lay Lab for letting me run this experiment and the Nachman Lab for providing wire-derived inbred lines. And I've recently started my own group at Arizona State University. So I'm looking for students and postdocs. So please feel free to contact me. Thank you.